organizers for giving me this opportunity here. It's great to be able to drive an hour and meet all the world's experts in collective dynamics. So today I'm going to talk about collective dynamics in swimming E. coli bacteria. And as you have heard from this morning's talks, collective dynamics plays important roles in many aspects of life. And it's either in a colloidal suspension, which is a non-living system, and or the living system. And so the mere fact that we are here, a group of people who have share same interests, gathered in one room, is a result of the collective dynamics. So I'm going to further enhance that by giving two examples of collective dynamics. One example at the molecule level and one at the cellular level. So on the left here, you see a movie of life of bacteria in about 60 minutes. So the bright dots here are the receptor clusters that are endow the bacteria to have a chemotaxis function. And in this part of the talk, I will talk about how receptor and receptor actually coordinate each other to form this chemotaxis function. And on the right here is the swimming bacteria. So these are E. coli, which are the bacteria that living in our gut. And non-pathogenic, so my biology collaborator said you can actually drink it. I never tried it, but it's very safe to work with them. So what's different from the usual bacteria living in your guts are the ones, these guys are fluorescent. So we put fluorescent plasmid inside so that we can look at it. So you can see that they are very dynamic. They swim around, and so when I looked at this picture at the beginning, I thought, well, they got to be chemically or mechanically communicating each other, and it would be a wonderful system to look at how mechanical interaction influence the macro scale behavior of bacteria. So the first part of talk, it consists of both experimental and theoretical work. So all the experiments are done by Eugene Coli, who's a postdoc in my lab. And our theoretical collaborators are Yuhai Tu, who is at IBM, and his student Lili Jiang. And so the basic approach that we took to look at this problem is a multi-scale approach. As you can see, this is to be universal approach in this conference now, so I don't really need to make an emphasis here. So basically we look at from molecule scale up to cell scale and scale up to millimeter, which we call macro scale. So why E. coli? So E. coli is something I view as a perfect model system for engineers and physicists. So here is a cartoon of E. coli, and so when I look at it, it's almost like a micro robot or micro machine. So it has its own sensory system here, which is the receptor cluster that you saw in the early movie. And so these receptors forms cluster because they interact with each other, which I'll talk more later on. So when they bind to the chemicals in the surrounding area, it sends a signal to this protein called TA, say, make a conformational change. So this is the way bacteria responds to the surrounding. So the conformation change of TA will facilitate the phosphorylation of this diffusible chemical called protein called TY, which then go on turn the flagella left or right. So the flagella turns either clockwise or counterclockwise. So the internal machinery is very simple. It's basically the main thing is this TA in active state, which is conformational change. You can think of it like flower open up, and the inactive state, where is a different conformation state where that it basically inhibits phosphorylation of this PY. And so what is remarkable about this is not only that it has a sensory system, responding system, it also has an adaptation system. So it's just like our eyes. When we go out in the sunlight, our iris turns smaller because it's too bright. So the same thing happens in bacteria. When there are too much activity going on in PA, the same thing that facilitated PY to dephosphorate also facilitated phosphorylation of the adaptation protein, which tuned down the sensitivity of the bacteria. So it basically has everything an engineer wants. It has input-output control and adaptation. So this organism has been a model system for chemosensing for about quite many years, about 30 years now. And so two things that is important to remember is one is high sensitivity. It can sense very little, low concentration of chemicals in the environment. And the second is it has a wide range of sensing, which means it can sense a wide range of chemical concentration. So here is a photograph rendition. This all this is not from the lab to show how bacteria moves. So when the flagella rotates counterclockwise, it moves the bacteria forward. When the flagella turns clockwise, it jams. And in this case, the flagella flies apart and the bacteria tumbles. So when you put bacteria culture on the microscope, this is what you see. And so here you see bacteria moves around, and you also see these rings. So these are the rings that is caused by lens aberration. And usually biologists don't like this because it makes the images don't look as pretty as they should be. However, for physicists and engineers, this is another thing that we can actually use to probe more information than what we can get from a normal microscope. So it turns out that this ring size is monotonically dependent on the Z position with respect of the bacteria with respect to the focal point of the microscope. So we can use the ring size to calculate the Z position of the bacteria. So for the image like this, instead of 2D image, we can figure out XYZ position of the bacteria, and as a return, using the usual tracking mechanism we do in fluid mechanics, we could find out the trajectories of the bacteria in 3D using a normal fluorescent microscope. So these software are now posted on the website. If anyone is interested in using them, feel free to download them. So if you look at the close-up of these tracks, you can clearly see these the run, turn, run, tumble characteristics. And here are two images I took from Howard Berg's publication, in which they made the flagella fluorescent. And so in this case, you can see that when the bacteria is making a run, the flagella bundles up in one bundle, and dynamically it's going counterclockwise. And when the flagella flies apart, the bacteria tumbles. And so these are the two states that bacteria has. So bacterial chemotaxis is a classical problem. Many people have worked on it. So this is a movie that's taken in my lab in a microfluidic device. So what we are seeing is the bacteria is sitting in a chemical gradient with a higher concentration towards the right, a low concentration towards the left. So at the behavioral level, Howard Berg in Harvard has figured out 30 years ago that the mechanism that bacteria do chemotaxis, which is that they actually lengthen the runtime when they feel that the chemoattractant concentration is becoming higher and higher. And they do nothing if the chemoattractant is decreasing or is not changing. So this has been figured out 30 years ago. And during the past 30 years, many work has been carried out to look at the signaling transduction pathway that's responsible for this chemotaxis behavior. So this map is also quite clear now that what is going on, how does bacteria, at molecular level, how does bacteria sense, what protein does the bacteria use to sense, and then translating into the motor protein. So that's also worked out quite nicely with many kinetic constants measured using Fret experiment. So basically, the key issue here is there are 5 receptors that are responsible for bacterial chemotaxis. And 2 of them are the most abundant, which has a copy number close to 20,000. And these are minor receptors, which is close to a few hundred receptor numbers. And so we basically looked at these 2 receptors, TSR and TAR, which are the most abundant receptor numbers. And so here are the contribution to these pathways, mostly from Howard Berg in this part of sensing and control part, and Philippe Cruzel in the motor kinetics part. So, what we have, as I said, that many people have worked on this field, and it's really not a good place for me to do a review here. So I'm just going to say that what we come in is really to use the advanced tool, which is our imaging technique and our microfluidic device, to make a missing link which to connect the molecular scale knowledge into the behavioral scale knowledge. So the experimental setup we have is agarose gel-based microfluidic chemotaxis device. So this is a device that we have developed specifically for bacterial chemotaxis. And the idea behind it is very simple. We take an agarose gel membrane about 1 millimeter thick, and we pattern 3 channels, parallel channels on it, and we flow chemicals in one channel, buffers in the other. Because of diffusion, there is a linear gradient established in the center channel. And so here is an image taken by flowing fluorescing in one channel and buffering in the other channel. So you can see that the linear gradient is established across all 3 channels because of the fact that the agarose gel, which is the material we use for this device, has the diffusion coefficient very close to water. And so the center channel is the place where we see the bacteria, and the bacterial migration towards the chemoattractant is imaged by the microscope. So another additional advantage of this is we can make many devices on this one chip. So usually we have 4 devices on one chip, and as a result we do 4 experiments at one time. And which saves a lot of time, which is actually quite important. We discovered that there are a lot of issues that if you do experiments today and tomorrow, they are not entirely reproducible. So it's kind of critical to do 12 experiments in one day so that all the data points in one plot actually make sense. So that part we are still twiddling on it. We think we know what the problem is, but for cell biology experiments, it seems to be very critical to be able to do many experiments at the same time. So high throughput using microfluidic device seems to be the way to go. So we are further we had further luck with a dynamic equilibrium I call. So when we look at this steady state, a steady state movie. So this is the movie that's taken after about 8 minutes. The chemoattractant has flowed into our channel. So by then the chemical gradient is in a steady state. And we find that the bacteria chemotactic process is actually is an equilibrium state, in the sense that the chemoattractant wants the bacteria to go to the right side, but the diffusion due to the density gradient wants the cell to go to the other direction. So as a result, we get a dynamically steady state, which is quite important for us because the data is quite noisy. So in this case, we could take 500 images and average them, and we would be able to get some useful information, quantitative information out of these images. And so this is the usual way we do our data analysis. We track the bacteria, and from these tracks we can figure out what is the density profile look like. And so this is the chemical gradient. So here, this is the good luck I was talking about, the balance between diffusion due to density gradient and chemotactic migration. And so from this, if you solve this equation, it's an exponential decay. So the decay constant will provide us with the chemotactic drift velocity. So this drift velocity is one of the parameters that we use to characterize how sensitive bacteria is. And the other parameter we use as the chemosensitivity measure is chemotactic migration coefficient. So it's basically the average Y position of all the cells. So Y is like this in this direction. So if CMC is 0, it means the cells are uniformly distributed in the center channel. And if it's 1, they all migrate to the chemoattractant side. So that's basically a second measure of what chemosensitivity is. So this just to prove that we indeed get equilibrium state. So this is the CMC as time, and you can see that with time, CMC changes. So CMC is the chemotactic migration coefficient, and it reaches steady state at about 8 minutes. So first thing we did is we looked at chemosensitivity curve of both two receptors that I talked about, the most abundant receptor, PAR and PSR. And we find, so the dots are the experimental data, and the lines are theoretical data. And so we find that the bacteria has a very wide chemosensing range and with a higher sensitivity. So in order to explain that, it is not sufficient just to use the classical kinetic theory, which says that the receptors don't interact with each other. Each receptor is an individual entity. And so the chemosensitivity is governed by the rate change of the ligand-receptor binding. So if that's the case, then this is the rate change of ligand-receptor binding, then it would be proportional to delta L over L. And this basically tells us that if we plot chemosensitivity versus delta L over L, which is the plot I showed you in last plot, then we would have a very narrow concentration region. And this high sensitivity region is going to be close to KD. So for example, for receptor TAR, KD, an unmodified receptor, KD is around 18 micromolar. And so this obviously cannot explain the experimental data we had where our sensitivity extended to more than millimolar. So the question is, how could bacteria do chemosensing with such wide range of sensitivity. So the questions started, the answers started to become clear towards the end of, towards 1998, 1999, where people start to look at the actual structure of the receptor. And they find through the cryo-electron microscope, and they find that these receptors actually they cluster together. And so this is one cluster. It's a transmembrane receptor. So here the membrane is here, inside the membrane, where they do their work of binding with the ligand. And here is the controlling site, which adaptation site. Sorry, which is we call methylation site. And here is the TA, which is the one that makes conformational change that facilitates the phosphorylation of TY, which is the motor. So the idea is that the fact that these receptors are all connected down here, the PA activity change might influence the receptor cluster in a cooperative way. So that's basically the key point that that the bacteria has such high chemosensitivity. So there are so this is the one of the early papers. There are actually quite a few theoretical calculations on the point. So this one is the first one that to propose that maybe the receptor goes active or not inactive is just like the spin, the magnetic spin goes up and down in a magnetic field, and the ligand binding is just like a magnetic field. So this group, Dennis Bray's group, basically hypothesized that the receptor a receptor was in the inactive state. In fact, it influenced its neighboring receptors through this PA conformation, and such that it influenced the neighboring states such that they all align to the same direction, which means they are either both all active or non-active. So here the dark is inactive and the white is active. So UHI is our theoretical collaborator. So next two slides is all UHI's work. So basically, UHI took the idea from biochemistry. So in biochemistry, there is what's so-called a MWC model, which says that the conformational state of regulated proteins are determined by the thermodynamic equilibrium. So this is well established in biochemistry, but it really hasn't been used in the setting where, like, the way that physicists use, like writing down Hamiltonian. So what UHI did is he wrote down Hamiltonian for the different states. So the energy state is illustrated by the cartoon here. Suppose you have two receptors. One is inactive, so A equals 0 is inactive, active is 1. And when the ligand binds to both these receptors, it makes the active receptor increases free energy. So this is caused by the kinetic constant change in inactive and in ligand binding and non-binding state. And same thing happened that when the methylation group put a methylation state on the inactive receptor, it lowers down the inactive receptor's energy level. So taking into this consideration, UHI wrote down the Hamiltonian for this system, which is basically this is the energy of active receptors, and this is inactive receptors. So the coupling that I talked about, how the receptor is influencing the neighboring receptor via conformational change of this PA, is included in this. So this this is less than 1, so it tends to align all the neighboring receptors in one state. So, so one could, the theorists in the room know much more than I do in this case. So what he did is he used the mean field theory for this Hamiltonian. And so there are also a little twist here. The methylation is happening at a much slower time scale as the ligand binding and unbinding. So in this case, this is millisecond and this is minutes. So what he did is he separated these two. So this Hamiltonian is just for one methylation state. And he calculates for the total 5 methylation states. And so he used the mean field theory, and so you can get an analytical function of the activity, which is this A here. So this activity of PA can directly be translated into the clockwise or counterclockwise motion of the bacteria. So when we took this and calculated the density profile, that is the density profile of the bacteria in a linear chemical gradient, and this is what we get in experiment. So in the experiment, we fix the average concentration of the chemical attractant and increase the gradient. And so you can see that the cells become more chemotactic as the chemo gradient increases. And in this case, we fix the gradient and change the average concentration. So the reason for this exercise is to test one of the hypotheses, which was also proposed in 70s, that bacteria actually sense concentration at a logarithmic scale. So if it's at a logarithmic scale, then they should go down with the average concentration and go up with the gradient. So this is, in fact, indeed the case. So these data, as I said, because of the microfluidics and all the tracking, is in fact precise enough to be able to reveal the fact that the decay constant, which is proportional to the average drift velocity, is actually linearly proportional to the gradient of the concentration. L is the chemical concentration. So this is, again, it's nature's way of managing a wide range of signaling. So from this experiment, we learned quite a lot. We thought that it looks like from this picture, it looks like it doesn't really matter whether it's TSR or TAR. Receptor doesn't seem to matter too much. So we thought that maybe receptor number is the only controlling parameter for the chemosensing. So we did a second experiment where we put a different chemoattractant into the two-sided channel, and one is the ligand for TAR, which is one receptor, and then the other is a ligand for the other receptor. And interestingly, we find that so this is the CMC. So this is for historical reason I call it chemotaxis index. And so the message in this plot is that what matters is actually only the receptor number ratio. If the bacteria, so in this case we control the receptor number. We put the receptor plasmid in the system. So what happened is when the bacteria basically sits there, the numbers of receptors on its membrane. If we decided I have more TAR, it would just go towards the where TAR ligand is higher. And then when the receptor TSR is larger, and it does the same thing. So it really doesn't matter which receptor it is. So this is quite gratifying for physicists. And there are some level of universality underneath these biological problems. So I'm going to switch gear to go from chemical to mechanical. And in fact, the collective dynamics in bacterial suspension, when I started to look at this problem, I always thought it's chemical. So we actually did. This is actually part of the reason we did this chemosensing experiment. Yes. Okay. So, so it turns out that in the specific bacteria that we use, that they move so fast that the diffusion of the chemicals is too slow for us actually to see an effect. So the result is, at this moment, I think there are still interesting problems there, how chemical sensing among bacteria influence its migration. However, for this specific E. coli, we find that the only interaction we have observed is hydrodynamic interaction. So this work mostly is done by Tian Liao, a postdoc in my group, and a few undergraduate students. And Cassiapi is a graduate student here with Don Koch, who's a theorist. And so Cassiapi is going to give a poster on more details of this problem. I only have 4 minutes this week. So basically, the E. coli motion. This morning there are a few. I shall talk about asymmetry lead to motion. So this is another example of asymmetry lead to bacterial motion. So basically, bacteria imparts its force to the fluid in an asymmetrical way. The reason is because the force around the head is imparted in a volume of a size of about 1 micron, while the backward force is distributed along the tail, which is about 6-7 micron. So this asymmetry basically lead to the first order fluid flow to be first order of force that's imparted to the fluid to be a dipole. And so Dom had looked at, write down the modified Stokes equation. So everything is the same because it's low Reynolds numbers. The only contribution here is the force exerted by bacteria to the fluid. So luckily, this stress term is quite similar to the polymer, except in the bacteria case, it pushes fluid for outward. In the case of polymer, it contracts. So you just need to flip aside, and a lot of things that have been done in polymers can be used here. So the first experiment we did is we did the pair velocity correlation, and so Don used the theory that I proposed in the previous slide, and so it fits reasonably well. So this is our longitudinal pair velocity and the normal pair velocity, and in Don's case, the theory is still 6 times below our experimental data, and now Don's they think that they have a way to fix this. So one of the interesting things that we find in the correlation is that the correlation starts to become significant around length scale about 10 micron. So these are longitudinal and normal velocity correlation. So we thought that if this bacteria is communicate with each other around 10 micron, then we could vary the bacterial density and to see what is the transport properties of the bacterial suspension. So we measure the diffusion coefficient as a function of cell density and find that indeed around, so this volume of 10 10 to 9 cells per meal is corresponding to about cell-cell distance 10 micron. So indeed, this shows up. So we think this is because of the bacteria as a gem. So this dip doesn't show up in theoretical calculation. So I'm going to skip this problem here. So I'm going to leave you with one of the most recent developments in my lab. So it turns out that a lot of things we learned in bacteria can be translated into mammalian cell chemotaxis. And the mammalian cell chemotaxis play important roles in many physiological problems. And I listed a few here: development, immune cell, tissue formation, cancer metastasis. And so the question is, how can we use the tools that we have developed and the ideas we have developed in bacterial chemotaxis and to look at some more sophisticated system. So here I'm going to end with this movie, which is the dendritic cells moving in a gradient of chemokine. It's a lymphoid chemokine. So you can see that these cells are very dynamic, just like a bacteria, and they form cluster, and they migrate readily towards where the chemo concentration is higher. So the question is, does the receptor in these more sophisticated cell line, in this case is dendritic cell, do they talk to each other, and do they have an adaptation system? So these are the range of things that we are working on. Thank you.
I'm not sure we can open this talk, so let's try that.
Maybe I missed the point in the second part. So you show that in the second part, the velocity correlation.
Right. Right.
Did you show that actually this hydrodynamic
Let's come out.
Yeah, we want to put it down a bit because title is not coming on.
Just take it down, down,
Okay.
down.
previous slide, you
Oh, it
made
go
the
up.
point that
Yeah, just do that.
moving
down.
from the
Right. So what happened is because the fluid here is moving outward. So when a bacteria moves next to it, it feels the fluid flow.
Right. So what is the graph on the right? You actually measure those points?
Yeah. So the dotted points are the measured points. And then using the modified Stokes theory, you can calculate the correlation function. So we plotted these two together.
It works.
Yeah, not quite. So the theory is 6 times lower than our experiment. So my collaborator Don and his students think they know what the problem is. But it's still progress.
What? This file format?
Yeah.
Some zip file.
Yeah, but why isn't it unzipping? Why am I not seeing the file?
On the next slide, you showed the diffusion of bacteria as function of
That's what I
density.
was trying to do.
Yes?
Not
Yeah.
So it's already unzipped.
So
Where?
you said that
What
when
is
you
it?
increase the density, at some
It's
point
again unzipped.
where the density is
It's a
such that
folder
the
or
average
file?
distance between
It's a file.
bacteria is
It's
about
a
10
file.
microns, the diffusion is the largest. Yes?
Right.
So I don't know how to do that. Let's try it here. Minimize this.
Do you have some intuitive explanation of
Yes,
why?
yes. So this is like in contradiction to Asha's result in the morning, right? When he has more of these chains, the diffusion coefficient decrease. In our case, it's increased. So this is entirely because when suppose you have a very dilute system, the fluid, the interaction doesn't show up because they are too far apart. Because the fluid flow, the fluid flow that's induced by the swimming bacteria is in vicinity of it's short range. So only when they are close enough that they can
What it has opened,
feel
just
the
15
flow
minute talk.
that is
But
created
that's the
by
type
the
that you want
bacteria.
excluded.
Then they can.
From where to
They so according to this, they like to trail each other, and so this is entirely. We haven't seen it in the real experiment yet, but it's entirely from this correlation.
No,
They like
PowerPoint
to
cannot
trail.
open this type of file. Okay, close it. Now,
So,
on that.
I have a question. Did you try to vary the gradient with time? It looks like your setup can easily do that. What do you think will happen there? You will get additional information from time varying gradients.
how many slides are there?
Time varying gradient.
This
So
is
there
only
are a
what
lot
you
of
start
work
from.
on time varying gradients by
Can't convert into PDF as well.
Howard
So for
Gross
raw
lab.
volts, you need to keep the
So
camera there
they
on the slide.
actually,
Okay,
yeah,
so
they
we can't
compare
open this.
temporally. So
Okay, forget about it.
But we have to
I
change
have
tape
not-
also.
Yes,
Okay.
We have to change tape by moment.
Very nice work. I was wondering how statistically significant this thing is. If it was due to hydrodynamics, I might have expected diffusion coefficient to maybe increase linearly or quadratically with the cell density. But you suddenly seem to have a spike and otherwise more or less constant. Is that because the statistics is not much?
Well, so there are things that we have not taken into account, like rotational diffusion and running motion to leads. So that could be one of the factors. So
I think let's take the speaker again. So our second talk is actually from the first talk from a real biologist in here. So Roy Welch, his PhD is from University of Wisconsin-Madison. Then he moved to Stanford to his postdoc with Dale Kaiser, and he's been in Syracuse after that. And he was going to be talking about genomics of emergent.
You again close the camera. No, please. This is also not working. No.
There we go. So we can get this thing working. Ready to go.
Your partner is connected.
Okay. First of all, this is actually the first time that I've ever given a talk at a conference where the university was sort of my home site. So I'm used to sort of being jet lagged and sitting in the back and feeling exhausted, but now I get to watch all of you feel that way, and actually I didn't have to get up until 8 o'clock this morning. But I would like to thank, first of all, Christina Marchetti for asking me to maybe even be a small part of organizing this. I was surprisingly nervous preparing this talk. I'm used to giving talks in front of microbiologists and sort of doing the same microbiology dog and pony show, and